Our last speaker for this session is Gerald Schatten, and the title of his presentation is Genome Editing Optimizations, Implications for Pre- and Post-Implantation Genetics.
Professor Schatten is director of the Pittsburgh Development Center, professor of statistics and gynecology, reproductive science, cell biology, bioengineering, and director of the Division of Developmental and Regenerative Medicine. He's currently president of the UNESCO's International Cell Research Organization and doctor honoris causa from the University of Nova Gorica, presented by the President of the Republic of Slovenia.
Thank you, Tony. Thank you, Jean. It's a great pleasure to be here, and the work that is going on is just so spectacular. What I would like to discuss today is where we are going in the future, and I'm hesitant in discussing this because there's part of me that would like to say this is the distant future. And yet we've already seen in the Ukraine and in Mexico, there are 3-parent babies. And in Pittsburgh, where I live, there is a cooperation between Uber and Volvo, where we have already driverless cars that will pick you up and take you wherever you want to go. And there's a temptation to say, oh, you know, this isn't going to happen for a while. And yet it's happening right now. And for most of my talk, I'm going to discuss a paper that came out 3 weeks ago from Shukrat Militipov's lab called Correction of a Pathogenic Gene Mutation in Human Embryos. But let me tell you, I'm going to talk about CRISPR, and I brought something that's very crisp. What you're seeing here is that there is an ancient system that was developed by bacteria all over the world to be able to read the DNA of viruses and inactivate, destroy the viruses. And it's a system called CRISPR, where there is a scissor-like enzyme called the Cas9, and it learns the DNA sequence of these viruses in the bacteria. And it can do one of two things. It can open up the DNA and then break it so that it edits it and destroys it, which means it's gone, but not repaired. Or alternatively, you can add a template and using homologous recombination, have that template read and not just destroy the gene, but replace it with a healthy gene. So that's going to become important in a second. So what you're seeing here is the DNA being recognized. Now it's being opened up, and there are these molecular scissors in the Cas9 that are breaking the DNA, and if you don't have the correct template, you're going to just get random repair and most likely destruction of the active gene that's non-homologous and replacement. So this will lead to the breakage being repaired, but with just randomness, and so it's in effect inactivating the gene. That kind of makes sense? On the other hand, it's possible, so it's just unraveling. It's possible to introduce a template of the corrected gene. So now you're not just removing the damaged gene, you're repairing it with the correct sequence. Don't think that this is theoretical. This is like Uber driving driverless in Pittsburgh. It is here. Oh my God. So for example, it's possible with dogs to destroy a myostatin gene and get double the muscle mass. I already spoke about how many groups are trying to model neuropsychiatric disorders and neural development using Cas methods to fight the ravages of Zika. People have made Cas-modified mosquitoes where the males are infertile and released gajillions of them out into Miami and other places where they were concerned about Zika. And you can say you have all these regulatory processes until people freak And they figure, oh, screw it, just let it out. And there are many other uses of the CRISPR technology. For example, it's too bright, but people have designed phages with the CRISPR technology that will kill pathogenic bacteria in mice and in humans. And so it's a way of developing an alternative to some of the antibiotics that no longer work. There are clinical trials already where patients have their lymphocytes modified so they don't have HIV/AIDS receptors on them, right? And so in a sense, you become HIV/AIDS immunized. Similar ideas are out there for various other viruses. Duchenne's muscular dystrophy, cystic fibrosis, all these other diseases theoretically could be treated using the CRISPR technology. And there is something extraordinary about CRISPR that is a little bit like an IRS loophole. This erases a bad gene. So it doesn't count as GMO. It's not genetically modified in the sense that you have to report that there's the new gene, because apparently erasures are different than insertions. And so already you're seeing mushrooms that won't brown, and these mushrooms and also apples and potatoes are deemed exempt from the USDA because it's just erasure, right? I mean, it's like getting a Vatican dispensation or something. I guess Jews don't know about that. Yeah, I mean, mice have been repaired. The cost of, you can't see the bottom of this, of CRISPR technology for dengue and Zika is now 61 cents for an assay that used to be a fortune. So it's totally revolutionary. And this leads us to designer babies. There are problems with Cas9 CRISPR technologies. And a paper came out in June showing that there was an awful lot of off-target problems that didn't show up in cell cultures, but do show up in animals. So for example, the predictive algorithms seem to do a good job in cells, but not in off-targets in animals. 2 independent studies, they had sustained more than 1,500 single nucleotide mutations and more than 100 larger deletions and insertions. This is not a good idea if you want to make a baby, right? I mean, even our genetics gurus know that. And researchers who aren't using whole genome sequencing are finding off-target effects, maybe potentially missing mutations. So let's say we want to repair this misspelling. If I'm only looking for the misspelling in the gene and it's ripped up, I won't find it. So it's gone. But you don't know that I've converted it from the misspelled word to the correct word, unless you do the sequencing for the correct word. So much of the work that's done is looking to see if you can find the mutation which you've removed. But it doesn't mean that you've corrected it, right? So that makes a difference. This paper that came out a couple weeks ago is a smart paper because they started with a man who had a hypertrophic cardiomyopathy. This is an autosomal dominant, so half of his sperm will have the mutation. Next, what they did is so 50% of the fertilized eggs will have the mutation, the other 50% won't, right, because half is half. In previous work from 3 different groups in China, they made fertilized eggs, they introduced the Cas9 after the sperm was introduced, and they ended up with mosaics. Now, obviously, having mosaics for a defect, especially a devastating defect that's going to lead to sudden cardiac failure, is not a healthy situation. They tried to do this instead by introducing the CRISPR with the sperm during ICSI. They then expect the paternal gene to be corrected, and later they found uniform, largely mosaic, largely non-mosaic, uniform corrections. And this is an important result that by introducing it early with the sperm, you can end up with embryos in which you're not getting the destruction of the gene, but rather the homologous repair of the gene. And let me tell you a little bit about DNA repair for those of us who are not experts in DNA repair. Using the CRISPR method, you can end up with the non-homologous end joining, which just takes double-stranded breaks and fixes them, but fixes them in a non-functional way. Alternatively, you can use this homologous directed repair when you have the correct template. Now, something remarkable happened in this paper. Instead of, they found the homologous directed repair, but something miraculous happened. They claim that in 41 of the 42 embryos in which they repaired the defect, it didn't use the corrected version of the gene that they put in, but rather it used the maternal copy of the gene. That somehow in that first cell cycle, the paternal chromatin found the maternal chromatin and used that as a template. And that's really quite remarkable, and it's a source of serious interest and concern. If it is possible during the first cell cycle for the paternal and maternal chromosomes to see one another and line up, and for mom to say to dad, hey, buddy, you're not cutting it, I'm gonna, fix you up here, God knows it's necessary. But that's a new version of genetic repair that has never been seen before, and it may be correct. But there have been criticisms in the literature that are appearing because in order for that to be shown, you need more evidence than they provided in this paper. And one of the reasons to think that they may not be correct is that when an ICSI sperm is brought into a human egg, the choreography of disassembly or of pronuclear development is slowed down, so that acrosomal regions remain on the sperm after it enters the egg, and parts of the sperm membrane that should be lost to the egg membrane come in, and there's even a yarmulke-like thing that stays on the sperm, and DNA synthesis won't start until after this cap comes off. And that's shown here in this animation, so that the choreography of the first cell cycle of ICSI is different than of IVF. So you normally aspirate the sperm into the needle. For those of you that don't like the Clintons, I used to use the verb suck, but since they are out of office, I don't do that anymore. Anyway, the sperm comes into the center of the egg, and the whole way in which it decondenses is totally different than if it had entered in the natural process of fertilization. And as a result of that, it's unclear really when the sperm DNA and the egg DNA get together. They probably get together at a much later stage than a slower pace than they would in natural fertilization. Okay, so Dieter Egli and George Church have come up with a number of concerns, and they have a paper online that describes that. And some of the concerns deal with this business about the homologous directed recombination, and was that really directed by the maternal chromosomes or by the introduced gene. Also, that the pronuclei really don't come together because they're separate as a male and a female pronucleus, and that then also delays things. And so there are a few problems in this: the efficiency, the mosaicism, the off-target edits, and whether or not they really removed the, they repaired the defective gene. We also don't know if there are going to be anomalies, because they brought these up to blastocyst stage, but this is obviously not ready to attempt in babies. The National Academy of Sciences is already issuing guidelines that this should be studied, but only for pre-implantation work. But we need to be careful, because the world moves at a very rapid pace. There's evidence that it's not that many dozen genes that are associated with human intelligence, and people may be pushing it. You can already see clinics which are claiming that you can choose the IQ of your baby. And if you want a baby with a 70 IQ, if it's a woman, 32% of them will have illegitimate children. I don't know about if it's a man. 30% will live in poverty. In contrast, if you want 130 IQ, now I'm sorry for all the physicians in the audience, and I'm sorry for all the scientists. 130, you become an attorney or a chemist. You know, you guys are chopped liver. But people are marketing this. This is crazy. This does not exist. And yet there's money and drive. And I mean, okay. There is another technique that I want to spend a couple minutes on that also is theoretically available for correcting genetic defects. And this comes from the oncofertility world. So in the top left, you'll see an oncologist talking to parents who are worried about their prepubertal boy, who is going to be treated for cancer, but in so doing, he may be rendered infertile. So they extract the spermatogonial stem cells from the boy. He ultimately is cured. He comes to the University of Barcelona, and as a Catalonian, gets a wonderful medical degree. And then later, the spermatogonial stem cells are reintroduced into his sterile testis. And you can see, because there's a balloon at the bottom, that everybody's happy and he's having his biological children because his fertility has been restored. This has also been done with a number of animals. This has not yet been achieved in humans. We achieved it in monkeys, where we took spermatogonial stem cells, and it's the work of Kyle Orwig as well as with Shukat Militopov. You can take the spermatogonial stem cells from a monkey, freeze them down, inject them into the testis, and here's a laboratory where we're injecting the spermatogonial stem cells into the testis, and if you notice, the men look very worried. Women seem not to have the same sensitivity to this. But using ultrasound, you can inject it into the testes, fill up through the ready testis, get the whole testis filled with these modified sperm, and then restore fertilization, and you can see the sperm and also the embryos that result from that. So theoretically, if a man wanted to correct his sperm, he could do it that way, or perhaps even put those spermatogonial stem cells into an animal model that would repair the genetics. Now, I want to wrap up by noting that CRISPR is a very fast-moving technology, and already groups are modifying every different part of the CRISPR molecules so that they're optimizing it for different purposes. And maybe, can we have the sound?
Mucus presses down on cilia inside the lungs, preventing the cilia from beating and clearing the mucus. The cause of this problem is an abnormal CFTR protein created from genetic mutations inside the cell nucleus. Our goal is to replace and repair the abnormal DNA sequence to create a functional protein and restore normal mucus. One way to do this is to use gene editing technologies that cut out and replace defective parts of DNA, such as CRISPR Cas9. The CRISPR Cas9 repair system has three components: a guide RNA that seeks out and binds to a specific location in the DNA, the Cas9 nuclease enzyme, a type of molecular scissors that cuts the genetic sequence at the precise binding site, and a repair template containing the correct genetic sequence for the gene. Once these three components are introduced into the cell, the guide RNA binds to and activates the Cas9 nuclease. Now, the Cas9 nuclease, the molecular scissors, can search the cell's DNA for potential targets. At a potential target site, like the one shown here, Cas9 unwinds a section of DNA to see if the guide RNA sequence matches. If the DNA sequence matches the guide RNA, the Cas9 nuclease cuts the DNA to create a break in the strands. To replace the mutation with a normal sequence, a repair process is used that precisely edits the DNA. For this editing process to work, a short piece of DNA template that contains the desired normal gene sequence is introduced into the cell. The cell uses this fragment as a template and copies the new sequence as it repairs the broken DNA. Once the first strand has been repaired, the template falls away, and the other strand of DNA uses the repaired strand to fill in the remaining gap, leaving both strands with the corrected gene sequence. The cell then uses the corrected DNA strand to produce a functional CFTR protein that allows the proper flow of salt and fluids through the cell membrane. Mucus returns to normal within the lung, allowing the cilia to beat freely and clear the lungs of germs and irritants.
So the Cystic Fibrosis Societies are already funding human work on this. There are a lot of clinical trials on this. The reason I'm spending time downloading these videos for you is to show you that there is global interest to bring it to the clinic, probably faster than many of us might anticipate, and in this global environment that we live in. If it doesn't happen in San Francisco, it'll happen in Bahia or some other place. Thank you for your attention, and if anybody wants a crisp apple.